Welcome to this lecture on dynamic programming in sequence alignment. If you have ever wondered how scientists compare human genomes, trace evolutionary history, or identify genetic mutations responsible for diseases, you are in the right place. Today, we will explore the mathematical engine that makes all of this possible. Let us dive in. To understand our topic, imagine trying to find the absolute best way to drive from New York to Los Angeles. Instead of calculating every single possible route all at once, which would take forever, you break the trip down into smaller city-to-city hops. Once you find the best way to get to Chicago, you write that down and never calculate it again. You just build the rest of your trip on top of that best path. This is exactly how dynamic programming works for DNA. We break the massive task of comparing long sequences into tiny letter-by-letter comparisons, saving the best score at each step. This turns an impossible brute force guessing game into a fast, efficient process. Now that we understand the basic idea, let us look at how it works in computational biology. Dynamic programming finds the mathematically optimal alignment between two biological sequences, like DNA or proteins, by constructing a two-dimensional grid, or matrix. One sequence represents the rows, and the other represents the columns. The algorithm systematically fills in this matrix by solving smaller subproblems. It relies on a scoring system that awards points for matching characters and penalizes for mismatches or gaps. By storing the optimal score at each cell, the algorithm avoids redundant calculations and guarantees that the final path traced through the matrix represents the absolute best possible alignment. Building on this matrix concept, the most fundamental application is global alignment. This approach forces two sequences to align from their very first letter to their very last. The specific method we use for this is called the Needleman-Wunsch algorithm. It is governed by a specific mathematical rule called a recurrence relation. This rule tells us exactly how to calculate the score for any given cell in our matrix, based on the cells we have already filled out. Let us look at the mathematical representation of this rule. Here is the elegant formula that drives global alignment. Conceptually, it looks at three possible moves to reach the current cell and strictly chooses the one that gives the highest score. The term f of I, j represents the optimal alignment score at the current row and column. The first option inside the max function represents a diagonal move, where we align two characters and add the match or mismatch score, s. The second and third options represent moving down or right in the matrix, which introduces a gap in one of the sequences, so we add the gap penalty, d. If we increase the gap penalty to a very large negative number, the algorithm will strongly avoid creating gaps and prefer mismatches instead. Before we move on, let us see this with a concrete example. We want to align two short DNA sequences, GAT and GCT. We are given a match score of +1, a mismatch score of -1, and a gap penalty of -2. First, we set up our matrix and initialize the first row and column. This represents the cost of aligning the sequence against pure gaps. Since our gap penalty is -2, the first row becomes 0, -2, -4, -6, and the first column does the same. This creates a heavy penalty for starting an alignment with massive gaps. Now that our boundaries are set, let us calculate the score for aligning the first G with the first G. We check our three directions. The diagonal move comes from the top left cell, which is 0, plus our match score of 1, giving us 1. The move from above is -2 plus the gap penalty of -2, giving -4. The move from the left is also -4. The maximum of these three options is 1. So we place a 1 in this cell. This tells us that starting with a match is the best possible first step. Skipping ahead to the final cell at the bottom right, we compare the T in GAT with the T in GCT. The diagonal score from the previous cell is 0 because of a mismatch earlier, plus the match score of 1 gives us 1. The other paths yield lower scores due to gap penalties. Therefore, the final optimal global alignment score is 1. This positive final score tells us that despite the mismatch in the middle, the sequences are reasonably well aligned overall. So, we have seen how to align entire sequences. But what if you are reading a massive, boring encyclopedia and you only want to find a specific, exciting paragraph about black holes? You do not care about aligning the entire book word for word. You only want the highly similar regions. This is the essence of local alignment. Instead of forcing two entire sequences to line up from end to end, local alignment searches for the best matching subsegments hidden within larger sequences. It is like finding a needle of similarity in a haystack of genetic data. Why is this targeted search so important? Local alignment is arguably even more useful in practical biology than global alignment. Often, two completely different proteins might share a small, identical functional domain, like a binding site for a specific molecule. If we used global alignment, the massive differences in the rest of the protein would completely drown out this tiny similarity. By using local alignment, scientists can discover these crucial shared domains. This helps them understand the function of newly discovered genes and how completely different organisms might share specific biological mechanisms. To achieve this local search, we use the Smith-Waterman algorithm. Notice the crucial addition of the 0 in this formula compared to the global alignment version. The other terms still represent the diagonal, up, and left moves. However, if all three of these calculations result in a negative number, the max function will select 0 instead. This means the score at any cell can never be negative. Whenever a score becomes negative due to accumulated mismatches or gap penalties, the algorithm resets that cell to 0. This 0 reset mechanism effectively cuts off bad alignments and allows the algorithm to start fresh, ensuring that only highly conserved, positive scoring local regions are highlighted. Let us see how this 0 reset changes our matrix. We want to find the best local alignment between ATCG and TCC. We will use a match score of +2, a mismatch score of -1, and a gap penalty of -2. Because we are looking for local alignments, we do not penalize the start of the alignment. Therefore, the initial row and column are filled entirely with zeros. This mathematically allows the alignment to begin at any character in either sequence without carrying a heavy burden of gap penalties from the unaligned prefixes. It is the mathematical equivalent of saying, start fresh right here. Now let us look at aligning the T in ATCG with the T in TCC. The diagonal move from the top left cell is 0, plus our match score of 2, giving 2. The other moves involve gap penalties and yield lower or negative scores. Since 2 is greater than 0, we place a 2 in this cell. This positive number indicates that a strong local match has successfully begun. We are starting to build our highly conserved local region. To truly understand the power of this algorithm, consider aligning the A in ATCG with the T in TCC. The diagonal move is 0 minus the mismatch penalty of 1, giving -1. The gap moves give -2. Because all calculated values are negative, the max function selects 0. The cell becomes 0, effectively ignoring this mismatch and preventing it from dragging down future alignment scores. After filling the entire matrix, we scan for the absolute highest number anywhere, which represents the peak score and exact location of the best local alignment. Here, we see a completed dynamic programming matrix. Notice the sequences along the top and the side, and the scores calculated inside each cell. The blue arrows represent the traceback step. Once the matrix is filled, the algorithm traces back from the highest score, following the arrows to reconstruct the actual alignment. Every diagonal arrow represents a match or mismatch, while horizontal or vertical arrows represent gaps. This visualizes exactly how the algorithm guarantees finding the best possible biological relationship between the two sequences. Let us summarize what we have learned. Dynamic programming is a transformative mathematical tool that makes sequence alignment computationally feasible by breaking the problem into a grid of smaller decisions. We explored the Needleman-Wunsch algorithm for end-to-end global alignment and the Smith-Waterman algorithm for finding specific local matches. By systematically calculating and storing the optimal score at every step, we guarantee finding the best alignment. This elegant matrix-based approach forms the absolute foundation of modern bioinformatics and genomic analysis. You now understand the core engine driving genetic comparison.